happy to have today with us Dr. James Koltes. Dr. Koltes received his bachelor degrees in dairy science and genetics from the University of Wisconsin-Madison and PhD from Iowa State University in genetics. Prior to studying at Iowa State in September of 2017, he was a faculty in the Department of Animal Science at the University of Arkansas. Among his awards and honors, he was a Fulbright Fellow to Belgium, where he worked in the lab of Michael George. At Iowa State University, he has two main research focus areas. First, investigation of new tools like sensors and biomarkers for use in the genetic improvement of feed efficiency in dairy cattle. And second, annotation of genome function as part of the functional annotation of animal genomes FANG consortium. Dr. Koltes also serves in a leadership role where he supports research capacity in data collection for animal genomics researchers and supports a shared bioinformatic and data science resource for livestock breeding and genomics researchers in the United States. Thank you for being here with us today, and the floor is yours.
Thank you. Appreciate the invitation and the introduction today to speak at the seminar for University of Guelph. As I get started into my seminar, just make sure, there we go. Can you see my next slide okay?
Yes.
Good, thank you. Before I start, I need to thank the real people who did the work. I'm lucky to be the coordinator and the ideas person and getting the money to do the work, but these are the folks that really put the time in to get everything done. So, you know, we've got my PhD student, Corey Sabursky, Corey Sabursky Cooper now, who originally started in my lab as a master's student and has been here with me since the beginning. She's done a lot of the work I'm going to talk about today. Leo James was a master's student and is now a PhD student. Mary Sue Mays, who kind of oversees everything. Abby Jancy, you'll see some of her work. We've had a host of postdocs and visiting scientists, and then lucky to have 14 undergraduates over time to help collect the feed intake data. Obviously, that's a very intensive process. This was my group from 2021. We actually do an internship and they have a project and we talk about the research and they'll do unique projects along the way as part of that internship. So we're going to talk about a couple of different research topics today, but I wanted to give a little introduction of myself. I know some of you, but you don't all know me. So I'm an assistant professor at Iowa State, as it was mentioned. I started in 2017. Most of my work that you're going to hear about today is sort of in the genomics and phenomics of feed efficiency in dairy cattle, although we're going to start doing some methane work that I will touch on just a little bit at the very end of this talk, looking at trying to create breeding values and understand new indicator traits for those very valuable and interesting traits in dairy cattle. And then I also work on the bioinformatics side for those that have used animalgenome.org, the animal QTL databases, ontologies, genome browsers, worked with different maps for SNP chips, genome build annotations, things like that. A lot of that comes through animalgenome.org. And then through FANG, I won't talk a lot about that work today, but we're hoping here we're going to start doing some new work in that area to kind of harvest and reuse the FANG data to make good use out of it to understand where our regulatory regions are in the genome in the future. My appointment, that's always kind of an interesting discussion with folks in different places. I'm 65% research, 30% teaching, and 5% service. The service, of course, the students may be less familiar with, but that's everything from serving on committees in the department for things like how do you manage your finances, to serving on graduate student committees and working on things for professional societies like our multi-state NRSP 8, that leadership role that was mentioned that I do, and then I teach a number of courses, two that I lead, a dairy systems management capstone class for our undergrads, an introductory genetics course, and then I help in two different graduate courses in teaching genomics and teaching applied breeding, specializing in dairy. That's a little bit about me. We don't always get that in seminars, but I always find that's nice to know a little bit more about your speaker. So digging into the meat of this talk, everyone is probably familiar here that the dairy industry and a lot of our livestock industries are working to continually find ways to improve sustainability. And so, of course, we're trying to use our genetic skills and our genetic tools to do that as well. The U.S. dairy industry has pushed for this carbon neutrality by 2050. a lot of progress has been made in that area, and that's why I always like to say we're continuing to strive for sustainability, because as we're increasing productivity of dairy cattle, of course, we're increasing the amount of milk produced already per unit of input. We're diluting the maintenance. So even just since in 2017, the 10 years previous, there was a 20% lower carbon footprint just for increasing our efficiency of production. So I find that's interesting. This came from Jude Capper, this infographic on the right, and I like to share that with folks. So folks at Guelph, I know, are working on feed efficiency as well, and I'll mention that again in a bit. But without a doubt, feed intake is an important part of sustainability. So lots of different estimates, anywhere from 40 to 60% in terms of how much feed costs in production for dairy cattle. So improving feed efficiency is obviously important and an opportunity to continue to improve profitability for farmers. There's that aspect of sustainability as well. We can't forget livelihoods and people, but also to reduce the environmental footprint. Estimating feed efficiency obviously requires individual cow feed intake data, at least at the level that we're trying to do it, to do genetic selection, because we want to be able to cull cows and select bulls for breeding. That equipment to do the individual feed intake measurement is expensive. It requires quite a lot of labor. And so our big picture dream, and you'll hear this repeated a little bit in the introduction and throughout the talk, is that we're hoping some kind of precision livestock technology is going to help us as a lower cost way of acting as a feed intake proxy, maybe some measurement from a sensor, ideally something that was already on a farm so you could run very quickly to application with an existing technology. That may be a bit of a dream, but a place where we wanted to start. Why sensors and genetics? And so this is a little bit of a US perspective, which I think might be interesting. In the US, we've been hearing for a number of years now that it's important to start incorporating sensors as phenomics tools to measure new behavioral phenotypes or unknown phenotypes that we might be getting from that behavioral data for genetic improvement. So in 2018, there was a paper published in Frontiers in Genetics. It was the blueprint for USDA for funding research for US animal genomics projects. And that was brought on, the emphasis in that report to work with sensor data was brought on in part by the United States National Academies of Sciences, who published a scientific breakthroughs to advance food and agricultural research in 2019. I was a part of a group that led a white paper as well to try to advance the information about what could be done with some of these sensing technologies and this idea that these new phenotypes might be heritable and useful for breeding. And I just grabbed this quick snapshot, again, kind of to give you an idea where some of the emphasis came from in the US. So sensors and phenotypes from sensors and precision livestock farming will be important in the global effort for collecting phenotypic and genomic data for breeding and discovery programs. Combining large-scale sequence and varying phenotypic information from sensors into databases will be requisite to enable biology to inform breeding and feeding programs. That again is from our United States National Academy of Sciences. This comes from a committee on agriculture that included folks like Allison Van and others in the US that set forth priorities for what they thought was very achievable and underutilized areas of research in animal genetics. So that's kind of the background or the emphasis behind some of the research that you're going to see today. I'm going to give you an overview here of the topics that I'm going to talk about. So in the first study that I'll discuss, we'll talk about if sensors can act as indicator traits or proxies for feed intake and or feed efficiency in dairy cattle. And I'll actually come back to that subject in a later project within the presentation, digging in a little bit deeper. So first I'll show you sort of the phenotypic associations of sensors with feed intake, and then we'll get into the genetic components. Because a lot of these sensors were developed to pick up health events, we wanted to know how the sensor's ability to detect health events impacted the feed intake, and also how the sensor, if it was associated with feed intake, was impacted by illnesses and the interplay of that data in terms of understanding the informativeness of sensors as a proxy for feed intake. That's obviously an important consideration. Is all of the information coming just because an animal's sick and it's a proxy for illness, or are we capturing, for example, some other metabolic information about activity by collecting that sensor data? Then I'm going to talk a little bit about some other work. We're curious about some of the underlying biology impacting feed intake. So I'll talk about our measurement of complete blood cell counts, CBCs, red blood cells, and white blood cell types, their relationship potentially with feed intake. Most of that work, really, I'll tell you, was our characterization of if those different cell types were heritable. Their counts were heritable, sorry. And if we could identify genes impacting the variation in those specific white and blood cell types. Then I'm going to talk a little bit about a nutrigenomics study in our group that was conducted to look at the association of a feed additive with animal health as well as feed intake. While we were looking into animal health, we started to ask, were there other ways that we could try to understand some of the genetics happening and some of the biological processes happening underneath the hood that might be impacting feed efficiency. And again, animal health was kind of an obvious one. So we were involved in a study that was partially a nutrition study and a genetics study. So that's how we got into that line of research. And then I'll talk a little bit just briefly about other things going on in my lab that might be of interest to you, and maybe a little bit about what's next in our feed efficiency research. So I want to start out with a little bit of background, again, talking about feed efficiency and looking at sensors as biomarkers and some common terminology that I'll use throughout the talk. So in this talk, when I talk about feed efficiency, primarily I'm going to talk about residual feed intake. There will be a place or two where I might talk about a daily version of residual feed intake that we call ADMI. But for the most part, this is based on residual feed intake. So again, looking at the relationship between the predicted and expected dry matter intake and how far a given individual deviates from that prediction, which you can see here on the red line with the individual's cows observation plotted against this predicted value with the blue points. So what does that equation include? This is based on the Templeman et al. 2015 dairy science publication. It includes contemporary group information, which includes the herd and season, parity information, days in milk, the milk energy, so milk yield, fat, protein, lactose, metabolic body weight, and then body condition score. And we use RFI because we feel it's a better approximation to the overall net energy use of the animal. So with our feed intake data, this is a big project. So it takes lots of people to get the work done. And so there are collaborators from Michigan State, University of Wisconsin, Iowa State, University of Florida, and USDA ARS at Beltsville, Maryland. This is the Agile group. So this grant was led out of Michigan State to be able to collect all of the data that we needed. So we had an existing database of around 6,000 or 7,000 animals, and we're trying to add an additional 3,000 animals with the project that I'm primarily going to talk about today. It's expensive to collect this data, and it requires a lot of labor. So these studies, we had anywhere between 28 to 70 days of feed intake data across a previous project, and then the project that I'll discuss today. And the newer project primarily had data over 42 days. And the goal was to get over 10,000 cows by the end of this with contributed data as a database to feed into the genetic evaluation for the US. And as part of this work, we began collaborating with the Resilient Dairy Genome Project at University of Guelph. And so a lot of the feed intake data, the milk data, the things needed to calculate RFI have been shared with the university, and there's some reciprocal sharing of data there as well, which has made for a great international collaboration because this data is so important in terms of sustainability and understanding feed intake and feed efficiency. So a little bit more background then, what is this data being used for? What is the genetic tool? It's what we call PTA feed saved. And so PTA feed saved, I apologize if it's something pop up on my screen here. I don't know if you can see it, but I'm gonna make it go away quickly if I can. is a tool that is really combining maintenance energy in terms of body weight and body size, or what we call body weight composite, and feed efficiency as residual feed intake. So with this measurement, one of the reasons why it was selected was because larger positive values are more favorable, and that's easier for producers to understand. So if we see a more positive value, that means more feed saved, and for this particular EPD that we're using, it's more feed saved per lactation. The heritability of feed saved is 0.14. And another advantage of it is that it's calculated to be uncorrelated with other traits that are accounted for in RFI, such as milk energy, in particular milk yield, and body weight factors, which account for maintenance. One thing that we really want to try to do is to not be selecting for cows, for example, that are tall and skinny, or lose the ability to select for increased milk production. We would like to be able to select for cows that are more efficient per unit of feed consumed, ideally. So just to give one more visual quickly of what that looks like, here's two cows and their PTAs for feed saved. So this cow A has a negative PTA, so it is not favorable. It is less efficient versus cow B that has a positive PTA for feed saved at plus 400. So if we were just, thinking about it for picking parents and which one would have the more productive offspring, again, obviously take the difference, our basic animal breeding in action. So there would be a 500-pound difference expected in the progeny of these animals, with the progeny of cow B expecting to be 500 pounds more efficient per lactation than the progeny of cow A. And that can be quite a bit of savings if we think about, an average of 3 lactations. If that holds, that's a bit of an assumption, but that could be 1,500 pounds of feed per lifetime, pretty close to a ton of feed per lifetime. That's certainly quite a bit. So that's a bit on the breeding value that we're trying to develop, and so the U.S. is trying to be more sustainable, right? We want to breed for more feed efficient dairy cows. So let's come back to the precision technologies aspect of this. Why are we doing that? So we want to make our breeding values for feed saved be more accurate. Compared to our other traditional production traits, we don't have the same prediction accuracy. In fact, it's a fair amount lower at around, I don't remember exactly, but somewhere around 0.35 reliability on average. So it's a fair amount lower compared to a lot of our production traits that are more like 70% or maybe even closer to 80% for a young animal. So the idea was we could collect our genotype data, right? We're going to do genomics and use our phenotype and our genotype data to estimate our breeding values, maybe get some sensor data then also, which we hope is going to be informative, and develop equations, determine the statistical relationships between the sensors and various feed intake and feed efficiency traits, and hopefully find a way to combine that sensor data as a novel phenotype, as an indicator trait, or proxy, as you'll hear me say sometimes, but really to get an indicator trait that is correlated to feed intake that allows us to gain more information, and maybe gain that on a lot of cows in the industry where we might not be able to anytime soon measure commercial feed intake. So again, the goal being of increasing the accuracy of predicting feed efficiency by adding sensor data to our existing genomics data with feed intake, body weight, production traits as phenotypes for a future version of PTA feed saved. So that was like our big goal in doing all of this work from the beginning. I'm going to start to step through now the studies that were done at Iowa State. And so as part of this work, my team was leading a lot of the sensor research. So the next couple of studies are all based out of Iowa State as a part of this larger project. So one of the first things that we wanted to do was look at a number of different sensors and ask which ones might serve as proxies for feed intake. Because again, we were looking for more data to increase our prediction accuracy, and looking for cheap, readily available data from commercial farms, where again, hopefully looking at some sensors that are already on farms would help us, assuming that data from the sensor was adequate as a indicator trait. So the first study I'll talk about was by my PhD student, Dr. Corey Sabursky-Cooper, in Frontiers, which was published about 2 years ago now, where we looked at the associations of wearable sensor measures with feed intake, production traits, and environmental parameters. And just briefly, these different studies come at different times within the project, so the number of animals will change a little. But we have 340 cows for this study, with feed intake data that was collected, daily milk weights, daily weights and body condition scores twice per week, and then weekly milk component information, and then 4 different sensors, which included CowSense sensor, SmackTech rumen bolus, environmental sensors, and SCR ear tags. You'll note we had different numbers of these tags and sensors. The reason for that was that they came at very different costs. The rumen bolus that we used, because it measured rumen pH, was quite expensive. The tags that we used were something that was already on farm, so it was easy to have it on all of our cows. And then based on our experience with other sensors, we used more or less of some of those. As we evaluated our sensor measurements, we adjusted them for contemporary group, parity, and days in milk. And for this study, this is one where we used this daily expression of RFI, this ADMI, which was essentially dry matter intake adjusted for the same factors that are in the RFI equation. So when we worked with this data, and I know this table's a bit busy, so I'm going to summarize it in just a moment. We had a number of different measurements per sensor. So in some cases, 2 or 3. We looked at combining sensor measures within a sensor and modeling those to determine if they were associated with any of the different factors or variables involved in the calculation of residual feed intake, and then estimated essentially the association based on if it was that sensor was adding additional information or picking up an association after adjusting for all of those different factors. And so in that previous slide, the black filled in, shaded in bars are actually where we had statistically significant relationships at p less than 0.05. But just to summarize that a bit to make it a little easier to look at, we had 5 of the 7 different sensor measurements that were individually associated with feed intake after adjusting for the RFI variables. We found that activity from our SCR ear tag, which is the one we had the most data from, was only significant when we adjusted for rumination, and then it was highly significant. We thought that was interesting because we know rumination on these tags are predicted from activity data, so we think there's some relationships there between those 2 different measurements. The largest effect that we identified was for rumen temperature at almost 3.5 kilograms of dry matter intake per degree Celsius. Again, these are daily estimates. And then all sensor measures could account for variation in DMI even after adjusting for those RFI variables. So we were seeing some new information, some new association with feed intake, even after accounting for the data that we already routinely have. And then last, we considered measurements 2 different ways. We either averaged across the measurement intervals, and we had a couple of different measurement intervals for these different sensors, or we summed and did a daily total for the sensor measurement. And we actually found that the averages of the intervals from the whole day were more significant and had larger effect sizes than the daily totals. So that was something else interesting that we tracked and utilized in future studies. So after doing this study, we were curious, well, what about other sensors? And one challenge about looking at a sensor or a subset of sensors is that every sensor can be different. These different sensors are owned by different companies. We're generally not getting raw data. In fact, in the last study I showed you, we had one sensor where we could get raw data, and then all of the others we couldn't. That company where we got raw data actually doesn't exist anymore, so we can no longer get that information. But with a new study that we started, we realized that we had access to some historical data using these neck collars that are used as part of the milking system at Iowa State. And so we thought, well, these are a pedometer. We could look at their relationship to feed efficiency and use some historical data as well to get an idea of what the relationship of activity looked like with a different sensor and with a little bit bigger population. Because again, this data is expensive. It's hard to amass lots of data in a data set where you have both sensor measurements and feed intake. So in the collar study, this was a NEDAP manufactured sensor that was part of a Bomatic milking system. So neck mounted, as you can kind of see in the upper right corner. And we had, for this study, feed intake on 676 Holsteins, ranging in parity from 1 to 6, days in milk from 47 to 254, 33 different contemporary groups, and on average 55 daily records per cow. So pretty nice sized data set. The bigger data set, so on the right in yellow, these are cows that don't have feed intake, but the total amount of data that we had for this sensor. So almost 1,500 animals and a mix of Jersey and Holsteins. So we wanted to play around with that data a little bit as well. For those 1,484 cows, we had on average almost 2 years of data, not quite 2 years of data per cow. So we thought that might be interesting to look at some of the relationships between the sensor and maybe some other behavioral aspects. Focusing on the feed intake first, we again found an estimate with a positive relationship between feed intake and activity. So as activity goes up, feed intake seems to increase, dry matter intake. And it was highly significant. So we actually saw this same association across all 4 of the sensors that we looked at in the previous study as well. So that's interesting that we were seeing consistency across sensors. We were curious if activity differed by parity. So here you can see activity on the y-axis and parities 1, 2, and 3 plus. And you can see that over time, it does appear the older cows are a little bit less active. And there was a quite highly significant difference. We had quite a bit of data for this. We are also curious if activity changed with stage of lactation. So you can see activity on the y-axis and stage of lactation as early, mid, and late. And so you can see on the side here in the key what that corresponds to for days in milk. As you go from early to late, it appears cows do become more active later in lactation than early in lactation. And then, of course, the curiosity I had, if you've worked with Jersey cows, you know they're a bit different than Holsteins. And kind of as we expected, Jersey cows appear to be quite a bit more active than Holstein cows on average when you're accounting for parity effects and differences in days in milk and all that kind of stuff being taken out of the mix in the modeling. So just kind of more of a curiosity on my part. You can see that Jersey cow curling its tongue, which is also a very interesting Jersey behavior. So shifting gears, after looking at all of those things, we were also, thinking along the way, okay, these sensors were all developed either to help us detect if a cow or a heifer is coming into heat, if it's coming into estrus, or if there's an illness. And so, as I mentioned early in the kind of the overview of the talk, we wanted to look at the impact of those health disorders on that measurement, the signal from the sensor in its association with feed intake, but also quantify the impact of health on feed efficiency, because at the time we hadn't seen a study like that, and we had a pretty good sized population to work with. So in terms of estimating the effect of different health events, we did have to get a little creative in how we put our health events together. This is essentially the same data set as you saw with the milking collar project, plus our data that we collected in the current FAR feed efficiency grant from the very first study. So we had close to 700 cows, and you can see that whether we look at lameness, mastitis, or other, and other is more rare health events, things like respiratory disease or injuries or things like that that were conglomerated together. We're losing anywhere from 0.75 kilograms to almost 2 kilograms per day with the observations that we observed for animals with those health disorders. I will mention, in this case, mastitis is a very mild mastitis because we didn't have the ability to measure severe cases of mastitis, those animals would have had to have been removed from our pens with kaolin gates to go to a treatment pen where the milk was dumped. So these are really mild cases. When I've talked to my colleagues, and for those of you who work with mastitis, you know cows that get a severe case of mastitis are going to really tank in milk production, and they really tank in feed intake as well. I have colleagues at USDA here that are working on publishing some data that say, you know, it's like 40 or 50 kg, 40 or 50 pounds a day, which is pretty significant. We also wanted to quantify what that impact of health was at different stages, different parities of cows. And so in this particular figure, you can see dry matter intake on the y-axis, and then our different health events with healthy on the far left as you face the screen, lame, mastitis, and other as you go to the right. So our highest parity, 3+ cows, are in yellow at the top, and then second parity in red, and in black is our primiparous, our first parity cows. And not unexpected, we see much larger impacts of health events on those older cows. Of course, they're more physiologically mature, so those illnesses are hitting them much harder. They're having bigger drops in milk production as well. They're having bigger drops in feed intake. We're talking about 6 kgs per day at least in those third lactation cows when you go from healthy to the other category. So, you know, something else we wanted to know is, were our sensors giving us extra information beyond just picking up a health event? And so in looking at the summary of this research, you know, we concluded that mild illnesses, mild mastitis, lameness, things like that, have measurable impacts on daily feed intake. And we could think about ways maybe to adjust for that somehow in our feed intake models if it was desirable. We had 11 different sensor measurements in this study that we looked at that were associated with health. And so we looked at what happened when you fit those different health events in models with feed intake at the same time. And we found that sensor data from 5 of the measurements from the sensors could account for more variability in feed intake based on the AIC statistic when accounting for health events and other information included in RFI. So there did appear to be additional information beyond those health events. The health events were definitely impacting some of the statistical association and accounting for some of the effects that were being detected by sensors in their association with feed intake, but there was more information coming from the sensor above and beyond those health events, it appeared. In some cases, when we fit our milk yield and composition data, it did account for those health disorders. It just depended on the specific models, but there were some of the sensors where still, when we added the sensor on top of the milk energy sink data, the body weight, the other variables in RFI, and the health event, we were still accounting for more variability in feed intake. So it appears the addition of sensor data as an indicator trait for dry matter intake could be useful, especially when health data is unavailable, because you pick up some of that information and then you're also picking up some additional information, it appears, in terms of association with feed intake. So we've talked about a number of different aspects of why we might look at sensors as indicator traits, but really we need to know what the heritability is of some of these sensor traits and the genetic correlations to feed intake and other traits impacting feed efficiency. So that's the last chapter I'm going to discuss. This was the final chapter of Dr. Corey Cooper's thesis. And so this is kind of the mature data set, the Iowa State portion of this larger FAR project, where we had almost 600 cows between 24 and 278 days in milk, again with our other measurements, feed intake, milk components and weights, body weights and body condition scores that were collected weekly. We had 877 cows that were genotyped, that also had data from our wearable SCR sensor that had activity and rumination data, as well as pedigree information. And we had access to PTAs from CDCB to work with as well for those cows. So I'm going to do my best to keep the population sizes straight here, because for some of the next analyses that you're going to see, we used the larger population for heritability estimates, and then a smaller subset of animals for the genetic correlations, because we needed to have both sensor data and feed intake, which is a smaller number than the numbers that I just showed you. So we estimated heritability. We looked at both a genomic estimate of heritability and a pedigree-based estimate using the following model that accounted for contemporary group effects, point in lactation, that did include initially heifer and late in lactation and dry period, parity 1, 2, 3 plus, and then we fit either an A matrix or an H matrix, depending on if it was a pedigree-based or genomic analysis, and then included permanent environmental effect for the repeated measures. The pedigree-based analysis was done with AS-REML, and the genomic analysis was done in AI-REML F90. So looking at those estimates initially, the heritabilities were around 0.21, regardless of if we used the pedigree-based estimate or the genomic-based estimate. And so the numbers here are pretty similar. We're talking about just shy of 1,000 animals, and the genomic is just shy of 900 animals used in those estimates. For daily rumination, our estimates were, I think, a little lower than what's been published before at 0.14 and 0.13, but again, the estimates were pretty similar, whether we used pedigree data or genomic data. The next big question, obviously, was what's the genetic correlation between these sensor measurements and rumination traits? And something I didn't mention earlier, we looked at all of these different sensors, and the thought was, what sensor do we pick as a candidate to look at these genetic relationships with? And really it came down to, what sensor we could afford and had the most information on. And so that's really why we focused on this specific sensor. We had it, the farm owned it, and we were collecting feed intake data at the same time. So we had the ability to get the data most easily. And these are sensors that are on lots of cows commercially. So that's how we got to this specific sensor. Coming back to the genetic correlations, then we fit a very similar model. It was a bivariate model, though, in this case, with the same factors included in the model. We fit both a pedigree-based model and a genomic-based model, again with the same softwares. Using all of the data, most of the genetic correlations were not different from zero. And a big challenge here is that now we're talking about a subset of that larger population. So only about 477, just shy of 500 animals, where we had the sensor data, the wearable sensor data, and feed intake. Our genomic correlations were essentially not different from zero for anything. So I'm going to talk strictly about the pedigree-based estimates from here on out. We did find for our pedigree-based estimates using all of the data, a relationship with body condition score that was kind of low to moderate. For rumination, it was a negative 0.3 correlation and 0.1 for our activity data. When we looked at specific points in lactation, though, and we thought this might be important, most of our feed intake data was collected from cows in mid-lactation, so somewhere around 40 to 200 days in milk or so. And we still didn't really have as much data as we'd like. We had subset our data between peak and mid-lactation, so from 51 to 90 days, mid-lactation 91 to 200 days. We combined that data and looked at our relationships, our genetic relationships, and found something rather interesting. The correlation between dry matter intake and daily average activity was negative 0.73, and with rumination was 0.56, but you'll notice those standard errors are still pretty big. We didn't quite have 200 animals that fit that range, probably because we had a number of cows that started in these days in milk and may have gone on a little bit longer. So we definitely need more data, but this is something very interesting to us. The positive correlation, I think, makes sense. More rumination is associated with more feed intake. The negative correlation between activity and dry matter intake has been a bit puzzling to us. So we've been looking at another sensor, our collar data, and evaluating the heritability and genetic correlations with that data set, because we have around 600 cows with that data set, and at least initially, we appear to replicate this result. We're not quite sure what that means yet, but that data set has half of the cows that come from an earlier time point, so they're not included in this data set, It's an independent technology, and we're seeing nearly the same correlation. So we're still working on figuring out exactly what that means, but it's quite interesting to us. That really summarizes the feed efficiency work looking for proxies with sensors in my research. I'm going to shift gears and quickly tell you about a couple of other research studies that I have. I may go through a little bit quickly for time's sake to make sure we have time for questions. But I wanted to at least share some new data and some things going on. So we were also curious about the relationship of health and some molecular measurements of animal health in terms of how they relate to feed intake. And so one idea that we had is we thought we'd look at blood cell counts from red blood cells and white blood cells. This was another study by Dr. Cooper, and so we had about 450 cows, of which 258 were genotyped with the AFI 60K chip, with the same data as previously. But in this study, we also added the blood samples to measure 15 different complete blood cell counts. And so we fit a model where we accounted for illness, parity, contemporary group, days in milk. And we identified, not surprisingly, relationships between mastitis and some of the different CBC measures, log lymphocytes. There are a number of other health disorders associated with monocyte counts. Mastitis was associated with log white blood cell counts, and there tended to be a relationship between mastitis and basophils, as well as with health events in general as an overall category with MCHC. So with those data, we also looked at their relationship to feed intake. And so you can see our overall model up above. We're using those same variables that we did in our estimates of residual feed intake, and then added the individual CBCs. You can see we've got a number of different red blood cell parameters here that are statistically associated with feed intake, and then a couple white cells, neutrophils, and lymphocytes. This was something I was actually kind of curious to dig into, because I thought, just from a pure science standpoint, could we find genes and mutations associated with these different blood cell counts, and also estimate the heritabilities. Heritabilities for CBC measures have been estimated in pigs. We had a previous study in beef cattle. Some of these had been estimated in dairy, but a number of these cell counts and cell parameters had not. So we estimated heritabilities for these different traits, as well as repeatabilities. And they had a pretty broad range. Some things with very low heritabilities, like our log leukocytes at 0.09, and some things with quite high heritabilities. I don't know if I quite fully trust those at 0.78, since we're only around just shy of 300 animals here, but definitely some of these quite highly heritable, and some of them around 0.51, 0.6. We actually could compare to the previous beef cattle study, and these heritabilities were similar in Holsteins as was observed previously in Angus. So I mentioned I was curious. We wanted to look at if we could look at essentially doing a genome-wide association study between these CBC measures and see what genes might be involved and how much variation we could account for. Again, we know some of them are associated with feed intake. Obviously, they're associated with illness. That wasn't surprising. And some of the effects were pretty good size that we found in our GWAS, ranging all the way up to almost 9% of the overall genetic variation, for example, in the log white cell counts. And just to give you a brief overview of what some of this data looks like, not surprisingly, we did find a number of regions that were pleiotropic that appear to be associated with multiple CBCs at one locus, so on chromosome 1 and chromosome 6. There were also a couple of regions on chromosome 4. Some of these regions had some really interesting candidate genes. There was a knockout for one of the genes, and I apologize, I can't remember it off the top of my head, but that gene actually was associated with CBC numbers in mice, and another one was associated with changes in immune function in mouse models. So I think there's some really good candidate genes underneath some of these GWAS peaks. Again, we're looking at genomic windows here associated with the various CBC traits. There was also an interesting region on chromosome 21 that was pleiotropic for 12 different measures. And then just to show you that, so here's an example for log lymphocytes. You can start to see some of these pleiotropic regions on chromosome 1, on chromosome 4, and on chromosome 6, as well as on chromosome 21. And then just again in white blood cell counts, and in some cases, these measurements are moving a little bit, chromosome 4, for instance. But very interesting, and again, some very interesting candidate genes that are underlying these traits. So in my last little bit of time here, I'm going to tell you about some work that we did with a feed additive. I mentioned the feed efficiency study was a partnership between nutritionists and geneticists. And so while we were doing this, there was an idea to do a study to look at a feed additive that was thought to impact animal health. And I don't have time to talk about that whole study today, but it was statistically associated with changes in animal health. This is a phytogenic compound called digesterone dairy. We had 108 cows, 54 control and 54 treatment animals. They received the same amount of soybean meal per day, and one of the treatments also had a small amount, 2 grams of this digesterone dairy included with it. The product in this case does impact feed intake. In fact, the animals that take digesterone tend to consume a little bit more, although when you account for the illnesses that were occurring in the pen, this actually becomes a non-significant difference. Digesterone also appeared to increase milk yield and components, so increase in milk weight, fat, protein. It's increasing the energy corrected milk by about 1.5 kilograms per day, and it was a very significant difference. And then I mentioned briefly about the health, the interesting thing, and when we started this study, you know, you're always worried you're going to be biased. So when my colleague at the company said this may impact health events, I made sure to say nothing to anybody that worked for me. I just said collect the data, and at the end, we'll look at it. And we always were routinely collecting health data because of the sensor studies. So we did this, and when we found such significant differences in health events between the control and the treatment, you know, I was surprised. There aren't large numbers of health events here, but there are on the order of magnitude of around 16. So there is a pretty good number of health events that we were able to look at. So it did appear that this product actually did reduce the incidence of health events. Alongside of that, we thought it'd be interesting to do a genomic study to say what genes were being impacted by this phytogenic compound. So I didn't mention it before, but there are essential oils, things like oregano, cinnamon, and some other compounds. Of course, the company will tell you some of the active ingredients, but not the amounts, and not everything that's proprietary, but at least to give you an idea. So what you're seeing in the schematic, we looked over 3 different time points. The green here is the digesterone treated animals at adding this specific feed additive. And you know, we're interested again in what happened and how that related to that phenotypic relationship we saw with the decreased illness and increased energy corrected milk for animals, again, that had this digesterone product fed to them. And we were looking at just half of the data set here, so 58 animals. We looked at the CBCs, and we did see a difference in the white blood cell count. It appears that the digesterone animals have a slightly lower white blood cell count. We didn't detect any other cells that were different by diet or diet by time interaction. We did see with digesterone a little bit of an increase in monocytes and eosinophils, and basophils tended to increase at the mid time point, but only in the digesterone group and only at a suggestive level. So we think the cell content, the difference in those immune cells could be contributing to the differences in health events that we observed in the initial nutritional study that was conducted. But we wanted to look at gene expression differences to see if there were differences, if there were anything interesting happening there that might explain what's happening at the molecular level. And we accounted for the cell content in that so that we could look at what might be a response to the diet of an increase in the number of cells versus a change in the gene expression straight away. So in the next 2 or 3 slides, I'll use this notation WO and WI. WO is an analysis that looks at all of the gene expression where we don't account for the cell number, and WI, and I think I keep the same color notation, is an analysis that counts for differences in the cell number so that the difference in gene expression that we observed is only due to the gene expression differences, not the changes in the cell count. So WO genes are related to differences in cell count. WI are independent of changes in cell count. Those cell counts were accounted for in the model as a covariate from our CBC measurements in the lab. So as we look at those results, we contrasted across the digesterome treatment at the different time points and looked at the diet by time interaction. And we identified 99 genes that were differentially expressed independent of accounting for the cell type. So that may be just due to differences in cell type, and 45 genes that were differentially expressed independent of the cell type. After accounting for the cell type. So these genes appear to differ in a cell type independent way. Oops. And just briefly to summarize some of that work, when we looked at those time by treatment interaction genes, the 107 that were different due to treatment across both time points in the treatment group appear to be enriched for interleukin-4 gene pathway, and we're still doing some work on this. This is a manuscript in preparation, but we're quite interested in what that means and why that may be associated with changes in feed intake. So kind of bringing things to a close here, summing things up. My overall lab is interested in identifying phenotypic and genetic factors that impact dairy cattle feed and production efficiency, and so we talked a bit about projects today related to using sensors, trying to identify sensors as indicator traits for feed intake, and then some genomic studies trying to identify some genes that might be associated with feed intake with some subphenotypes. These are other things that I work on in my lab, either broadly related to feed efficiency within my group as part of the overall consortium with Mike Vandehar at Michigan State, and then some other things in my lab looking at metabolites, some of this nutritional genomics work with the phytobiotics, and then we've done a little bit of work on beef on dairy. I didn't get a chance to talk about the bioinformatics work, but this is the bulk of the dairy work that we're doing. And we're still working a little bit, digging into some of the fatty acids in milk and what are some of the genes underlying that. We've got an EQTL study ongoing, and then looking at the heritability and the correlations of these sensor traits. That's just being finished up now from the third study that I talked about today in my lab. So take-home points from the overall study. So Iowa State's contributed a large amount of data towards the PTA-saved genetic tool that the overall U.S. Feed Efficiency Consortium has developed to let dairy farmers select bulls and cows based on feed efficiency. And then at Iowa State, we've identified a host of wearable sensors that are associated with feed intake and efficiency traits in dairy cattle to add information beyond what we already use to estimate feed efficiency as RFI. Not surprisingly, I mean, we knew that health events impacted feed intake and efficiency, but we quantified it between 3/4 to 2 kilograms per day feed intake change with the 3 different health events that we observed. And we identified that our sensors do pick up effects due to health, but also additional statistic associations above and beyond health events when you fit those in the statistical model. So there's a host of different bits of information that those sensors may provide to help us learn more about feed efficiency, and they may be able to act as indicator traits. Our preliminary data indicates some fairly high genetic correlations Again, we need to replicate these things, but rumination with a genetic correlation of 0.56 between dry matter intake and activity from our SCR sensors, and minus 0.73 between activity and dry matter intake. I showed the CBC data. We found that it was associated with feed intake, a couple of the specific white blood cell types and red blood cell types. Some of those CBC counts had quite high heritabilities, and there were some interesting biological candidate genes that were identified. Will there be implications for use in genomic selection? I don't know. I don't know if those are really things we can select for, but I think some interesting biology there, and maybe a piece of the puzzle that will help us understand a little more how health and feed intake are related. And then in doing this last study, really as part of a large collaborative team looking at this phytobiotic compound, we identified the association between that specific additive, phytobiotic additive, CBCs, that it impacted, CBC levels in whole blood, white blood cell counts, and was implicated in pathways involved in inflammation and oxidative stress based on the gene expression data. And then finally, from some of our CBC work, metabolite work, and other things that are going on, we think there's a number of interesting pieces of information here that might help us better understand what's going on with feed efficiency at the genetic level. We're working on this EQTL study. I'm hoping that'll help us as we combine that data with our FANG data and other functional information to identify more genes, regulatory regions, and mutations that might eventually be useful for selection. That's looking out, obviously, into the future. So to close today, I have a number of folks I need to acknowledge, but most importantly, FAR and CDCB for funding. I mentioned the study funded by Biomin, which is now a part of DSM, as well as I need to acknowledge USDA for historical data for the collar study that I mentioned in the middle with the bomatic milking collars, and then some funding for some other projects here that I didn't get to talk about today. Coming soon, we were just funded as part of the Greener Cattle Initiative. This is a study actually led out of the University of Wisconsin to measure methane and to work on looking at the relationship of feed intake and methane and the microbiome with methane, and developing a genetic tool that might be useful for selection for dairy cattle in the U.S. With that, I want to make sure to leave at least a little bit of time for questions. Thank you for your attention.
Well, thank you very much for this amazing presentation. If anyone has questions, you can raise your hands, turn on your cameras and microphones, or you can also type the question on the chat. So yes, Lucas, you can go.
I don't know if you can hear me well.
Yes,
you can.
Yeah, hi James, really nice to see you again. Great presentation. There's a lot going on in your lab. I have many questions that we can probably talk about later on, but I think my main one is about the older cows that you were talking about being more hardly impacted by the health events, and that got me a little bit puzzled. But I wanted to ask you, what's the span of the effect on these cows? Because they might get affected more harshly at the beginning, but they might recover faster as well because they're more resilient animals in general. So did you get to look at that? And because in the long run, they might be harshly impacted at the beginning, but come back faster than a younger cow would. So do you have any information on that? And the other question I have, because you said that you were puzzled about was it ear activity and feed intake?
Yeah.
That were negatively correlated?
Yep.
Isn't it the case that animals that eat more often are not moving their ears at all?
I'm going to take that last question first. I haven't heard that before. That's new to me if that's the case. We do wonder if it's something related to the behavior. And with our kaolin gates, we can't per se measure that. We know how much feed they eat, but we don't know how often they're coming to the bunk. So it could be related to that. We do know, for instance, with lame cows, they tend to do what we call kind of binge eating, where they come to the bunk and will they seem to kind of gorge themselves and go away. So in that short
Yeah.
time period, they walk a lot, and then they'll be still for a long time, and they bob their heads a lot. So there are some strange things there. We tried accounting for illnesses. It actually made our genetic correlations even stronger in some cases. So we know it's not a health effect on that correlation, or at least not one that we've been able to track down. To your question, you're asking if cows that are sick earlier in lactation, so earlier in days in milk, if there's a bigger effect of the illness on feed intake?
No, not really. It's just maybe I understood your points wrongly there. But what I understood is that younger cows or older cows, let's say, get affected more harshly by health events.
Oh, yes. Yep. Later lactation cows do. Yes.
Yeah. So I was just asking, what's the time span of that effect if you have information on that? Because I'd assume that an older cow could get affected more harshly at the beginning, but will recover faster than a younger cow.
That's a good question, and I haven't looked at that directly. I wasn't surprised initially because older cows are at a higher level of production, and so when they get hit by an illness, it seems a lot of times the higher producing cows drop more in productivity anyway. So, from that standpoint, it seemed logical to me. But in terms of getting over it, that's a good question. I haven't looked at that.
Okay. That's it for me. Thank you.
Okay, thanks.
Thank you, Lucas, for a question. We may have time for one question if anyone has a quick question. So this is the last call, people. Okay. We have Lucas again.
Well, if no one's asking, I'll ask. The other thing I was thinking is more generally speaking about the use of sensors from commercial farms, and if it's been easy to convince more and more farms to make use of those, because when the farms already have those, it's easy. It's a no-brainer to keep them going with it. But how easy has it been to actually gather more farms, commercial farms, to join the projects?
Yeah, so that's a good question, and so that's something we haven't done yet. That's something we're investigating actually. Time will tell. I think how you recruit people into these studies is always a bit of an interesting topic. You hope you find folks that are alumni from your university that are collecting these data, or farmers that want to help. And something that I've learned regionally is, as I've moved around different parts of the U.S., there are places that I think folks are sometimes a little bit more supercharged to help out, and in other places where folks are a little bit more secretive. Everyone wants to protect their competitive advantage, I think. And so sometimes folks aren't quite as willing to share data, but at least in my experience, there's always been a number of folks that have been. So I'm hopeful as we move forward that people who have these sensors are willing to let us have it, because I think we have some very interesting results. But before we could say, an ear tag-based sensor is an indicator trait, we've probably got to have 1,000, 3,000, maybe more cows to be able to say there's a confident genetic relationship with not such a big error that we can use to potentially look at as an indicator trait. And then obviously we need to think about things like, how does that potentially impact the cow in other ways? And we've looked at associations with some traits, some of the other RFI traits, but probably need to look at relationships with other health traits and fertility traits and things like that as well, if we can.
Okay. Yeah, thanks, James. Nice seeing you.
Yep, thank you. Likewise.
Thank you, Lucas, for a question. I believe we ended now our seminar. I would like to thank you again, Dr. Koltes, for the presentation today. It was amazing, and hope to have you in the future sometime.
Sounds good. Thank you for inviting me.